I'm Silvia Marsoni, I'm Director of Clinical Research at the Istituto per la Ricerca del Cancro di Candiolo, Torino, Italy. And I have been there for a year and a half now. It's about primary and secondary resistance to target therapy in colorectal cancer, which was funded by ARC. It's part of the 5 per 1,000 ARC project, and these are large projects heavily funded by ARC to study in a comprehensive way all the translational research involved in a subject. And our subject is what I said, resistance to target therapy in colorectal cancer. We started by using cetuximab, the anti-EGFR monoclonal antibody, as a probe to understanding how patients respond and why the majority of these patients actually do not respond to EGFR anti-therapy. And we did so by growing a very large platform of what we call xeno patients. That is, these are fresh human tumor that are transplanted in mice so that the mice becomes kind of an alias of the patient, an avatar of the patient. And we have made the decision of growing a large number of these tumor xenograft, approximately 300 and counting, so that we can design and do clinical trial in the aliases. And then if something comes out, we can translate that into clinical trials in the true patient. And we were lucky because approximately 6 months into the project, we found out that a very small proportion of colon cancer patients, but a large proportion of those patients that are KRAS wild type, and therefore should be responding to an anti-EGFR therapy, and in reality are not responding, carry an amplification of the HER2 genes, which, as you know, it's been found as a driver both in breast cancer and in gastric cancer. So we did a little trial in the mice and discovered that by using a combination of lapatinib, a multi-tyrosine kinase inhibitor of HER1 and HER2, and trastuzumab, an anti-HER2 monoclonal antibody, we could cure the mice, literally cure the mice. So we brought this idea into the clinic, and I hope in 6 months from now I will be able to tell, we will do another interview and I will be able to tell you if our original idea was right, because we're going into the clinic in a month from now. Yeah, well, we're not doing just that. I mean, the project is a very large project that it builds on this platform, but also has a library of colon cancer line on which we do the pre-screening. We have approximately all the colon cancer line in the world we've banked. And then there was all the molecular profiling that goes along. Each of these tumor, once they are collected from the patient, gets profiled. We do have another milestone that looks at secondary resistance. So we have patients that have the primary tumor sampled and profiled, and then when they relapse, we have a second sample and we do whole genome assay of the two. And again, we have found things that are quite interesting. These we just found, so I cannot talk yet because they have to be published. But again, this is going to stem another clinical trial very soon. And we do have a large part that is concerned with MET, c-MET gene, because my institute is famous. The PI of this project is Paolo Comoglio, who is one of the father of MET. So we're looking at MET. And there is another big chunk of the project that is looking at stem cells in colon cancer. And actually, that's really kind of interesting because what we are doing, this is Carla Boccaccio. She's growing what we call colon sphere. And then we actually also transplant the colon sphere in the mice. and then treat the mice that have been transplanted with the colon sphere. So we got several models that are all honed down to study what is the primary and secondary resistance. Alberto Bardelli is one of the other guys that are heading the molecular part, and Livio Trusolino and Andrea Bertotti are responsible for the Xeno patient platform. I think that this is a very useful model because it gave us a true idea of what is going on. We can probe the mice with all kinds of things that we wouldn't be able to do in men, and then translate that to the clinic, because the sixth and final milestone of the project, which is headed by Salvatore Siena from Niguarda in Milano, it's the milestone that has to put into the clinic whatever we discover. So this is really hypothesis-driven, it's a machine for hypothesis-driven trials, which is what ARC wanted. I mean, and it's very in keep with what we are discussing here at the OACI meeting, translational medicine, personalized P4 medicine. Actually, we were very lucky. I mean, we're good, but we are also lucky. I mean, in science, you have to be lucky, because this discovery about the HER2 was done, we could have done it on, later on, and we would have come to the same conclusion. But obviously, having it done at the beginning, it carried up with itself a kind of a momentum that brought us to the clinic with a year and a half in advance of what the ARC wanted. All these projects have to have a clinical output at the end of the third year, and we are into the clinic now, a year and a half ahead. Yes, and we were lucky. Yes, this is the first time that the ARC, and I'm really grateful to them for doing this, has decided to a strategy of putting a lot of money into a group, into groups, and all the groups were judged. The winner were judged by a 20 people, fully international committee, and we were among the first 5 that were awarded, and the only one that were awarded in solid tumor. But it is very interesting, and I would like to suggest to ARC that we should have meetings of the consortium, because there are a lot of things that we can exchange one with the other. We already work with with Robin Foa group, because Giorgio Inghirami, who is in Torino, who is a pathologist in Torino, also has a Xeno patient platform for AML and myelodysplastic syndrome. So we do a lot of things together. For instance, we work on to do this kind of things, to do personalized medicine, you have to have an infrastructure which is really, really very cumbersome, money-wise, organizational-wise, quality control-wise. And therefore, it is a good idea. I totally agree with Shilsky today and other speakers that suggested that no single institution, nor even no single nation, can probably set up this kind of programs. I really worked 10 hours a day for the last year and a half just to bring back, bring up the biobank, organize all the stuff. You need a network of pathologists, you need a network of clinicians, you need a network of science, you need information technology up to here. There's a lot of organizational stuff that it's important. That doesn't make any sense that I do all this and then somebody else replicate that. We should put this effort together. And I think that ARC, if ARC supports this, it will make Italy go from here to there in terms of infrastructure in translational research. It's very different from the past. You could do very good science in your own lab. Now, working on with the clinic and from the clinic, you need to have an infrastructure that costs a lot of money, and especially a lot of thinking. And that's not, it's a loss of time not to share this. It's non-competitive, but at the same time, if you do not have that, you cannot be competitive science-wise or clinically-wise.